Imagine fighting for each breath. This is what cystic fibrosis patients have to face every day.
Continue blowing, a few seconds more, hang in there, push further if you can, hang in there, keep blowing.
Cystic fibrosis is an autosomal recessive condition that is the most common inherited disease to cause lethality in the Caucasian population. Its homozygous recessive inheritance renders carriers of the cystic fibrosis gene silently asymptomatic. 1 in 25 Caucasian Australians are genetic carriers for cystic fibrosis. This disease of the exocrine glands is multisystemic, but primarily manifests in chronic infection of the respiratory tract and causes associated pancreatic enzyme insufficiency. Death is principally attributed to end-stage pulmonary disease. The cystic fibrosis gene encodes for an intrinsic transmembrane conductance regulator, CFTR protein, that functions as a chloride channel. CFTR plays an important role in maintaining fluid balance in the lungs. Surface fluid is required by the lung in order to maintain both contractility and our defences against pathogens. Normally, CFTR will move chloride ions from the epithelium into the mucus, which causes water to move in the same direction via osmosis. Without proper epithelial chloride transport, the mucus will thicken, increasing susceptibility to infection and chronic obstructive pulmonary disease. Cystic fibrosis is a disease that has 6 different origins for the onset of its pathophysiology. Firstly, Class 1 mutations result in the inability to synthesize the CFTR protein. Class 2 mutations cause misfolding of the CFTR. Class 3 mutations produce a reduction in the functionality of the CFTR channel opening due to defective channel gating regulation. Class 4 mutations cause reduced chloride conductance. Class 5 mutations lead to a reduced synthesis of CFTR. Class 6 mutations result in reduced CFTR stability at the cell surface. Cystic fibrosis has no cure. Current treatments focus mainly on controlling symptoms for patients. Significant amounts of medication are necessary to address the variety of consequences associated with the disease. The main challenges faced are the complicated medical regimen and the lack of treatment designed to correct the underlying CFTR protein dysfunction. In order to address the disease as a whole, we propose that we re-establish an equilibrium between chloride and sodium conductance. This will be achieved by upregulating the calcium-activated chloride channel, TMEM16A, and downregulating the sodium transporter, ENaC. The protein HClCa1 can increase calcium-activated chloride conductance through undergoing self-cleavage to reveal its N-terminus, which can independently gate TMEM16A. Salarabanel's 2015 study demonstrated that cells exposed to HClCa1 will have increased surface expression of TMEM16A. TMEM16A is a functionally similar channel to CFTR, and as such can secrete both chloride and bicarbonate, which will restore the efflux of water in airway epithelium. However, it does not provide the same inhibitory action CFTR has on ENaC, which maintains an equilibrium of chloride and sodium conductance. N-acetylcysteine is currently being used as a mucolytic in cystic fibrosis patients, and it has recently been proposed that it functions by downregulating ENaC. Rochat's 2004 study demonstrated N-acetylcysteine downregulated the number of alpha subunits of ENaC present at the membrane surface. Our new proposed treatment for cystic fibrosis relies on a combined effort of both HClCa1 and N-acetylcysteine. The 2 drugs will be taken separately, both as nebulizers, with HClCa1 administered in the morning to increase chloride efflux, and N-acetylcysteine taken at night to decrease sodium influx. This dosing schedule mimics a normal physiological equilibrium. Unlike specific class-targeted medications, these treatments would assist all patients irrespective of the type of mutation, allowing us to bypass the predominant hurdle that has hindered drug development in this field. Our novel therapy will use a nanoparticle drug-based delivery system to move the pharmaceuticals into their target cells, specifically and efficiently. HClCa1 and N-acetylcysteine will be independently bound to a biodegradable polymer nanoparticle known as polybeta amino esters, or PBAEs, via encapsulation. PBAEs have been shown to successfully penetrate the nanopores of the mucus layer to reach the underlying epithelium. The nanoparticles will possess aptamers, which will guide the nanoparticles to the epithelial cells. The cis and lack of immunogenicity make them useful ligands. No signs of toxicity have been shown thus far, and this targeted delivery should greatly increase the efficacy of our combined therapy.
Imagine what this treatment could mean for sufferers of CF. We believe Includesco is the drug of the future for cystic fibrosis patients.